revenu, je fais ma présentation. Donc, vous pouvez avoir un peu des réflexions par rapport au travail. Et puis, une fois que j'aurai fini, vous vous mettrez en groupe et vous travaillerez sur cette petit travail pendant 45 minutes. Et puis, on vous ferait une petite présentation, chaque groupe, sur ces travails. D'accord? C'est bon? OK. Merci.
Thank you very much, Maria. All right, so I'm going to get into the first lecture for this morning. And as I alluded to yesterday, so I've given you an overview of different aquaculture systems. In that presentation already, I provided some information and context to these integrated systems in South Africa that we've been working on. I spoke a lot about abalone farming yesterday. So that will give you a good overview of the typical practices that are involved in farming abalone. And what I want to try and do today is zone in a lot more on these integrated abalone Ulver IMTA systems. And I'm essentially going to be presenting to you some case studies of research that we've been conducting within these systems, to try and get a better understanding of the functionality of these systems, what the benefits are of these systems, address some of the biosecurity concerns that have been raised for these systems. And Kevin spoke a lot about biosecurity and how reuse and recycling and use of waste can potentially be problematic. So a big focus of ours was to try and address that concern for the industry. And then present to you as well a sort of what we call a SWOT analysis. So looking at the strengths, weaknesses, opportunities, and threats associated with each of these systems. Sorry, I don't know what's going on here. The quality is really poor. What? Probably connect to. So we'll close the lines. Apologies for that. We will be with you now. That's right. Wow. You want better connect more from last time connect. Encore de Internet, peut-être? That's what I was asking. What network are you on? I am on, je donne Right, everybody. Apologies for that. So, as I was mentioning, what I'm going to do is I'm going to be presenting a sort of SWOT analysis, which stands for strengths, weaknesses, opportunities, and threats that are associated with these integrative practices. And actually, we did a SWOT analysis in 2009, so several years back. This was done by a very close colleague of mine, Professor John Bolton. So what I'm going to do today is I'm actually going to go back and I'm going to have a look at that SWOT analysis. And I'm going to go and see what was documented back then in terms of the strengths, weaknesses, opportunities, and threats. And we're going to see how those things have actually changed based on some of the research that we've now more recently done within these aquaculture systems. So to start off with, I'm just going to quickly go over the abalone aquaculture sector and kind of introduce the sort of evolution of the development of these integrated systems within the country. So the commercial industry essentially began in the 1990s. So this was after they had done some smaller scale experimental research in the 1980s already. But the industry only essentially began in the 1990s, and some of the factors that contributed towards this development was firstly when they showed that abalone could be successfully spawned in captivity. They found that the growth rates of cultivated abalone was much better than animals that are collected from the wild. And another key factor was the development of a market for what they called cocktail-sized abalone. So these are abalone that have a shell length of approximately 100 mils or a corresponding weight of approximately 100 grams. And these are animals that were dominantly sought after by people in the Asian markets. Now, since that sort of initial development, the industry has expanded dramatically over the years. And I think it's quite nicely illustrated in this graph on the right-hand side, where the green line shows total aquaculture production in the country, which you can also see has increased exponentially. But looking at that total production in relation to abalone production, which is shown by the green bars, you can see this exponential growth over the years. And in 2021, approximately 2,500 tons of abalone were produced. Now, at the advent of the development of this industry, the primary feed that was fed to abalone was kelp. And this is because abalone occur obviously naturally in these kelp beds that are situated along our coastline, but most of those kelp beds are located on the west or the southern part of the country to the west of Cape Agulhas, which is the most southern point on the African continent. So that sort of explains why they were mostly feeding that particular species of seaweed. But as I mentioned yesterday, this has largely been replaced by compound feeds for various reasons. The main one being to try and further enhance the growth rate of these cultivated species. But in addition to those farms that developed on this side of the coastline, there were also two other farms in the early stages that developed further along the east coast. And of course, these farms fell outside of the natural range of where Eclonia maxima, which is the main kelp species that they used, where that occurs. So this basically forced these farms, as I mentioned previously, to start cultivating their own seaweeds to feed to the cultivated abalone. And at that stage, they focused on two species. The one species was Gracilaria, which is a red seaweed. I'm not sure whether that occurs at all in Senegal. And then another species that I've spoken a lot about already, which is ulva, which is a green seaweed, which forms these large blades and is about 2 cells thick. But as they started cultivating these seaweeds, what they found was that they were having some problems with the cultivation of the gracilaria in these large-scale commercial systems. Essentially, what was happening was the gracilaria, as it tumbled around, got knotted up, and it also got caught up in these paddle wheels in those large D-ended paddle raceways that I spoke about yesterday. And due to that and a variety of other factors, they've actually moved predominantly towards the cultivation of ulva. So most of this ulva is actually grown on roughly 4 to 5 large commercial farms in the country. And these are some aerial photographs of some of these farms. I've already mentioned some of these in my lectures. One of them on the east coast, which is Wild Coast Abalone. That was actually one of the initial farms that were developed. And as I mentioned, this particular farm, because it fell outside of the range of the natural distribution of kelp, was pretty much forced to develop ulva cultivation at a very early stage. There's also 2 other farms in the Western Cape region, Irvin and Johnson Cape Abalone, which is located around Gansbaai, and then, of course, Buffeljags Abalone, which I'm going to talk a lot about this morning. In addition to that, there's one farm on the in the Northern Cape province, and that is called Diamond Coast Abalone. And that farm is actually a sister farm to Buffeljags Abalone. And then there's also another farm actually in the Western Cape called Abergold, which also produces some seaweeds, but not at the scale that you see in some of these other farms. Although there's a lot of interest, and this farm is actually starting to expand its production. So, as I mentioned yesterday, the production of the ulva in these large commercial farms is predominantly produced in these D-ended paddle raceway systems. So they are roughly about 30 meters in length, about 10 meters in width, and they're only approximately half a meter in depth. So they're pretty shallow ponds. And also the bulk of these farms are also growing this ulva in effluent water that is obtained from the adjacent Abalone raceway systems. So the production of ulva has actually expanded quite dramatically over the last 2 to 3 decades. And I think this aerial photograph actually illustrates that very nicely. So this is an aerial photograph of Wild Coast Abalone, which is on our east coast. And this was taken in 2002. And as you can see here, there was only roughly about, I think it was 10 ponds that were on this farm at that time. Sorry, I think it was around 20 raceway systems. And then this was a recent Google Earth image that I looked at end of last year. And you can see how this farm has expanded over the years. So they currently have more than 30 of these large D-ended raceways. And this is only on one of our commercial abalone farms. So myself and my colleagues were actually quite interested to try and work out what the total production is across the various farms in the country. So in order to do this, we made an estimate of what the annual production is per month. Obviously, that is going to change seasonally. So in summer, you're going to get a lot better production because you have a lot more sunlight. Most of our rainfall occurs in winter, so we have a lot of cloudy days. The production does go down a little bit, but on average, you're looking at about 1.5 tons of ulva that is produced in one of these D-ended paddle raceway systems per month. And from Google Earth images, we calculated that in total across the country, we have approximately 131 of these paddle raceway systems. So that works out to just over 2,300 tons per annum. So that's a pretty accurate estimate, and I think in line with what has been actually reported by many of these farms to the department. And again, like I said, this value will obviously vary across seasons. So what are the main drivers for this production? And I've actually touched on this a little bit already. The first one is the production of a supplementary feed for abalone. As I mentioned, it was a necessity for some farms where they had limited access to large quantities of wild seaweed stocks. And it was also shown to be very essential for feeding on these farms, particularly during the warmer months of the year. So at the peak of summer, when the water temperatures go up, a lot of these farms experience problems with feeding these compound feeds that were rich in protein. What happens is it causes bloat within the abalone due to the proliferation of bacteria in the digestive tract because these animals are stressed. And that caused mortality. So at those times of the year, the only things that they can feed are seaweeds. And another big driver for the development of these integrated systems is to allow for the recirculation of water. So farmers quickly realized that if they could recirculate water on their farms, that it would significantly save on pumping costs. And you can just imagine the cost that it takes to pump these large volumes onto these large commercial farms from the adjacent coastline onto the farm. And obviously that cost is going to vary on the head height of that farm. The higher that farm is above sea level, the more it's going to cost to pump that water. So if you can recirculate water using smaller pumps on your farm, that leads to a cost saving. They also found that if you circulate this water, you could get slight increases in water temperature, and that helps to facilitate growth of animals. And obviously, if you're growing these seaweeds in these ponds, you can ultimately use those seaweeds as a supplementary feed, and you can reduce your feed costs. Right, so like I said earlier on, Bolton et al. In 2009 conducted an initial SWOT analysis of all production in land-based aquaculture systems. This was essentially done to assess the strengths, weaknesses, opportunities, and threats of this practice to gain a better understanding of why this practice has been successful and things that could still be done in order to promote broader uptake of these more sustainable technologies. So when this initial SWOT analysis was conducted, at that stage, this was in 2007, just over 1,000 tons fresh weight of ulva was grown across the different farms. Most of this ulva was primarily fed to the abalone as a supplementary feed. And it was only later on that one of these farms started to recirculate the water on the farm. So, and that was to remove nutrients that dissolved in organic nitrogen from this effluent water before it was pumped out, and obviously also to save on pumping costs. And that was Evan and Johnson Cape Abalone, located in Park Spa. And at that stage, there were at least 4 species of ulva that were actually identified across the various farms. And those included ulva lactusa, ulva rigida, ulva capensis, and ulva linsa, which is actually not a very nice species of ulva to actually have in that system. And the only disease problem that was documented at that time was caused by a brown epiphyte known as Myrionyma strangulans. And basically what that epiphyte does causes little brown spots on the surface of the ulva. And as those spots develop, you start getting holes developing within the blades of the ulva. And eventually what happens is the ulva completely disintegrates within the system. So it's not something that you would ideally want to have in your system. And the main potential threat that was identified at that time was that some farmers were very wary of these integrated systems, fearing that they could promote disease. So feeding ulva grown in effluent water back to your animals, as well as the recirculation of the water with the systems, was considered a threat. So that prevented more uptake of this technology. So one of the primary strengths that were identified from this initial SWOT analysis was that ulva proved to be fairly easy to grow. The nice thing about ulva is that you can maintain it in this vegetative state within the system. And if you provide it with enough nutrients and enough light, it actually grows very rapidly within these systems. And there was a lot of literature already at that time that showed provided a lot of support actually for the production of that seaweed in that particular system. So growing ulva in land-based systems also provided a good crop for farmers at that time. So this obviously meant that there was greater employment opportunities. We also found that farmers were very adaptable at that time. So they were very keen on adopting new technologies and diversifying and trying to look at new species. So we also regarded that as a strength. Ulva grown in these systems, particularly in the abalone effluent, was also shown to have a much higher protein content compared to ulva that was collected from the wild. So in the wild, protein content of ulva is typically around 12 to 15%. But when you grow it in these recirculating aquaculture systems, that goes up quite substantially, at least to above 20% of the dry weight. And on average, what we found in those systems is that it ranges anything between 22 to 25% of the dry weight. And that can obviously then serve as a good feed. And there was also a lot of research coming out at the time to demonstrate that the use of mixed feeds of seaweed together with formulated feeds help to enhance the growth of these animals and to improve their general health. And also at the time, there was some preliminary data that showed that ulva grown in these systems had an impact on the microorganisms within that systems. So it wasn't very complex analysis that was done at that time. They were simply using colony forming counts to assess total numbers of bacteria. But in general, we found a reduction in those bacterial numbers. And I'm going to go a lot more into this, where I'm going to show you using newer tools for this, next generation sequencing, how we've shown how the microbiome is benefited from the ulva within these aquaculture systems. And then, as I've mentioned before, the growth of the ulva in these systems also enabled partial recirculation in these systems. And at that time, as I mentioned, there was only one farm that was doing that. And since then, 2014, actually, there are two new farms that adopted this technology, and they were continually recirculating 50% of the water within the system. The strengths that were suggested back then and identified back then, most of those still apply today. So the two farms that I just spoke about that more recently adopted that technology are Diamond Coast Abalone, showing you this earlier, as well as Buffeljag's abalone. On both of these farms, the system has been sort of set up as modular units. I think the one at Buffeljag's very nicely illustrates this. So on this farm, you've got what they call platforms. So this that I'm showing you here on the slide is considered as one platform, and it consists of 4 separate clusters with each of these platforms, and there are 7 platforms across this farm. And that was designed specifically to try and address biosecurity concerns that people had at the time. And Kevin spoke a lot about this. So there was no mixing of fomites, there was no mixing of staff or any other form of equipment, or no transfer of water between the different platforms. So if you had to have a disease outbreak on one of these platforms, the idea was that that would then be confined within that platform. And if you are practicing good biosecurity on your farm, you could then prevent the spread of this pathogen across the farms. So just quickly again, just looking at how these systems work. Basically what happens is that in each of these modular units, all the effluent water out of these abalone raceways, and there are a total of 42 tanks in one of these clusters, all that effluent drains by gravity into the adjacent D-ended paddle raceway system. The ulva growing in those paddle raceway systems will then extract all that dissolved inorganic nitrogen out of the water. The bioremediated water then drains into a sump at the base of that D-ended raceway where it is mixed with fresh incoming seawater, and the rest of it just flushes out back into the ocean. But remember, we've already removed most of the dissolved inorganic nitrogen, so it doesn't have an impact on the surrounding environment. And this mixed bioremediated water will then recirculate back to the adjacent abalone raceway systems. So as part of an EU-funded Stroll project, and I introduced Stroll yesterday, we did extensive research in these systems to get a better understanding of the physiochemical parameters in the system, and most importantly, to get a better understanding of the impact of the microbiome within these systems so that we could actually address some of these biosecurity concerns. And all of this research took place at Buffelshoek Abalone. So let's get into some of the data around the physiochemical parameters. So initially what we did is we looked at the physiochemical parameters in a 50% recirculating aquaculture system. So that was a standard practice across the entire farm. But what we also looked at was whether we could further increase circularity within the system. So we looked at whether we could increase recirculation rates up to 75%. For extended periods of time. And we also looked at whether we could have full recirculation in these systems for shorter periods of time. And I'll explain why we were interested in that in a few seconds. And in each of those systems, we monitored oxygen, pH, temperature, and the nutrients within each of those IMTA systems. And in all of those parameters, we observed these very clear daily rhythms, which is what you would expect in an integrated system like this, particularly when you are cultivating seaweeds. So we typically found higher temperatures during the daytime. Obviously, when your atmospheric temperatures are a lot higher, we also found higher oxygen concentrations during the day. And this is obviously when your ulva is photosynthesizing, so it is producing a lot of oxygen in the system. And we also, with regards to the abalone in this system, remember yesterday I told you that they are nocturnal feeders, so they're mostly active at night, and they're also feeding at night and excreting at night. We find that your pH levels and oxygen obviously decline during the evenings. In terms of dissolved inorganic nitrogen uptake, we found that in these large commercial systems, that up to 60% of the dissolved inorganic nutrients are removed as this effluent water moves through those D-end paddle raceways. And that's actually really impressive if you consider the large volumes of effluent water that actually moves through those ponds. In terms of electrical consumption, if we increase the circulation from 0 to 50%, we had a reduction in electrical costs from 10.35 kilowatts per hour per ton of abalone in these systems to 6.8 kilowatts per ton. So that is in one of these clusters. And if you calculate the energy saving across the entire farm, that works out to roughly 40% cost saving across this entire farm. Total ammonia and nitrogen and your free ammonia and nitrogen concentrations in that system tends to increase with the increase in recirculation rates. And as I mentioned earlier on, you also have this typical diurnal or daily rhythm in the concentrations of those parameters within the system. And like I said, that's directly linked to the activity of the seaweed and the activity of the ulva. But what's really interesting is you get very interesting relationships between all of these different water quality parameters. So particularly when I'm looking at nutrients and pH, what you find is that when your production of nitrogen in that system is highest, so early morning when the animals have excreted a lot of nitrogen into the water, your pH levels are actually at their lowest because of the high oxygen consumption and increase in CO2. And that actually helps to reduce your free ammonia nitrogen concentrations in that water. So that's actually quite a big benefit. But I think what I want to emphasize here is that these systems are actually quite intricate. So you need to gain a very good understanding of these systems in order to manage them correctly and ensure the health of the animals in the systems. So just to roughly summarize things, what this data indicated to us is that IMTA could feasibly be run for extended periods of time at 75%. So that could represent an additional cost benefit to the farms. But we also showed that 100% recirculation was also possible for short periods of time, around 3 to 4 days. And that's really, really useful. But as I mentioned yesterday, particularly if you have harmful algal bloom events, because that essentially enables that farm to shut itself off from that surrounding environment and protect itself from these blooms, particularly if they are toxic blooms. So just looking at the use of the ulva that is grown in these integrated systems as a supplementary feed. One of the initial things that we looked at is to see what the impact of that ulva would be on the overall consumption by animals on those farms. And generally what we showed is that even very small amounts of ulva that are supplemented into those baskets help to significantly improve overall consumption of feeds within those systems. So as little as 25% fresh ulva led to a 90% increase in overall consumption rate, which means you're making better use of the feeds that are included within those systems. So the other thing that we also wanted to look at is if we are growing all this additional seaweed on a farm, how much can we feed to the abalone within their systems and use as a substitute of the formulated feeds that are added to those animals? So how much of that formulated feed can we actually take out without compromising the growth of the animals within that system? So we ran extended growth trials, and what we did is we fed animals either a control diet, which consisted of a formulated feed, or mixed diets consisting of formulated feed and ulva. So in this case, we had 100% of their daily ration of the formulated feed with ulva, and then we started to slowly reduce the amount of that formulated feed. So 80% of what they should be getting, 60% or 70% of what they should be getting, all the way down to 40%. So taking as much as 60% of that formulated feed out of their diets. And as you can see here, we do not see any significant decrease in the specific growth rates of these animals or the condition factor of those animals. And in fact, you actually see a general trend of an increase in growth rates of animals that are fed diets supplemented with these seaweeds. So that means you can, and we could probably even increase this, you can take out as much as 60% of these formulated feeds out of their diets without compromising growth. So that's potentially a very huge benefit to these farms. All right, so as I mentioned, a key component of what we were doing was looking at the microbiome within these integrated systems. So this is a non-metric multi-dimensional scaling plot, and essentially this is just showing you the microbiome communities associated with each of the components of these integrated systems. With the top part of this figure showing you the microbiomes associated with these integrated abalone ulva systems. So this is systems that have been fed with effluent water. And the bottom half of this showing you the systems that were fed just with regular incoming seawater, so they were not receiving any effluent water. And you can clearly see distinct communities between the IMTA and the non-IMTA system, as well as the different compartments of these systems. So this is your effluent water coming into an ulva paddle pond, and that is your bioremediated effluent water and that is the microbiome associated with the ulva grown in those systems. So you can also see a very clear distinction between the microbiome associated with seaweed compared to the microbiome associated with the water itself. And as this water moves through these de-ended paddle raceway systems, you can see a shift in the community associated with that, where you can actually start seeing that the community associated with the bioremediated water slowly starts to reflect the community of bacteria associated with the ulva grown within that system. So the ulva is clearly having this sort of modulatory effect on the microbiome within that system. All right, so this is a plot of the relative abundance of the bacteria associated with each compartment in that system again, and this shows you the top 30 bacterial species that are associated with each of these compartments. And I think it's quite a complicated figure, but I think the main take-home message that I want to bring home here, just looking at all of these different clusters or colors, and each of those colors are representing different bacteria, you can clearly see that it's very high diversity associated with all compartments and with both the IMTA and the non-IMTA system. And this is a good thing because that generally is indicative of a healthy and a resilient aquaculture system. We also found that there was slightly higher diversity associated with the IMTA as opposed to the non-IMTA system. And this is possibly due to the higher nutrient levels that are associated with the system. And I think also sort of a maturation of the microbiome in these recirculating systems over time. Didn't find any specific pathogens associated with it in that system. And in fact, we actually found a lot of beneficial bacteria within these systems, bacteria that were important for things like nutrient cycling, as well as bacteria that were important to the growth and the health of the ulva that were cultivated within that system. And just something aside, if you're ever interested in ulva and you read up a little bit on it, you'll actually find that there are actually specific bacteria that are associated with that seaweed in the system that are very important for the growth and the morphogenesis of that seaweed. And if you remove those bacteria, the seaweed will actually just disintegrate within that system. So it won't develop into that typical layer. The other very interesting thing that we observed in that system, and I showed you already that you're seeing this change in the microbiome, but we found that it sort of modulated specific taxa, the abundance within that system. And one that I wanted to highlight here was vibrios. I think most of you working in aquaculture will know that there are very common opportunistic pathogens within aquaculture systems. And you can see their relative abundance as it moves through the system is significantly reduced. So in addition to what the ulva seems to be doing in the water and on the ulva itself in terms of the microbiome, we also looked at the benefits that this seaweed has when it is fed back to the abalone that are cultivated within the system. So again, we ran an extended trial and we fed the animals various diets. So they were either fed a formulated feed or fresh ulva, so those were the two controls, or we fed them mixed diets consisting either of fresh ulva, dried ulva, and some of the components of ulva. And for those components, we looked at ulvan, which is the primary carbohydrate within the seaweed, and we also looked at glucuronic acid, which is one of the building blocks of that polysaccharide, but has also been shown to be very important in terms of gut health. And at the end of that trial, we removed the digestive tract from animals within each of these treatment groups, and we looked at the microbiome associated with that. So again, I'm showing you a multi-dimensional scaling plot just to show you the overall communities and how they're clustered. And in this case, we actually observed a large extent of overlap of the microbial communities in abalone that were fed the different feeds. With abalone fed the fresh ulva clustered together with the other diets, but you do see that there's some separation of this microbiome of these ulver-fed abalone compared to the rest of the two groups. And when we looked at a Venn diagram, again, this shows that there's a large degree of overlap of the microbial communities across the different diets, but there are 18 genera that are not present in the gut of the abalone that are fed fresh ulver treatments. So again, showing you that the fresh ulver is having an impact on the microbiome of the animals that are consuming the seaweed together with their other feeds. Just quickly, this is again looking at the relative abundance of bacteria in each of these diets. Again, yeah, you can see there's a large degree of diversity. But if you had to compare the diversity associated with the digestive tract of the animals compared to the seawater and what we find in ulver, you will find that there is a slight reduction in diversity. And that, again, is something that you would expect because we're looking at a far more specialized environment. So you'll typically see that in animals. And I think the main point that I wanted to make here again is that we again saw distinctive differences and changes in the microbiome when we looked at animals fed either fresh ulver or the formulated feed. And again, yeah, I've highlighted the Vibrio group where you can clearly see a significant reduction in their numbers in animals that are fed the fresh ulver. And this is just one taxa or bacterium that I'm talking about, but we also observe this for various other bacterial taxa as well. All right, so just to come back now to Bolton et al.'s SWOT analysis that was done in 2009, and we're going to look at the weaknesses that were identified back then and see what still exists today. So there is this continued weakness for including for the low value of ulver on farms. Start down quickly. So most of this ulver, as I mentioned before, that is grown on these farms is predominantly used as a feed. There isn't really a commercial value that is associated with this. And because of this, many farmers don't really see the benefit in growing it on their farms. So I don't think they're considering all the other benefits that I've slowly started to highlight here. And that makes them a little bit reluctant to adopt this technology on the farm. It also still remains fairly easy for most farms just to feed a formulated feed. And in fact, as I've mentioned before, many farms still consider this to be the most biosecure method of feeding your animals on a farm. In addition to this, these systems still remain fairly novel to many farmers, even though cultivating ulver at a commercial scale on several farms in South Africa for more than 2 to 3 decades. And again, this obviously makes it problematic for these farmers to realize the benefits. Another thing is that infrastructure for growing a lower valued product is obviously considered to be high, and most farms will rather use that space that would have been used to grow ulver for cultivation of a higher valued species like abalone. But again, yeah, I think one just needs to take into consideration your site selection up front in order to ensure that if you're wanting to grow abalone in integrated systems like this, that you choose sites that will enable you to do that. Other weaknesses that were identified back then was the content of DMSP in the flesh of abalone that were fed ulver, particularly those animals that were canned. DMSP breaks down to dimethyl sulfoxide, and that gives it like an off odor and a taste to the abalone that are canned. But that is generally only a problem, obviously, when you are canning animals, and there has been more recent literature to show that you can actually depurate these animals, but that depuration process does take some time. Another weakness that was identified back then as well was there was very little information available on the identity of ulva within these systems, but we've done a lot of molecular and morphometrics on the ulva collected from all of these farms, and our recent studies have shown that in fact today there's actually only one species of ulva that is presently cultivated on all of our farms, and that is ulva laciniata. And finally, there's also been a lot more extensive research that has been conducted on the growth of ulva within these integrated systems, largely addressed some of the lack of knowledge around the optimal conditions required for the growth of species. So in terms of some of the threats within these integrated systems, such as concerns around the disease caused by this myrianema that I spoke about earlier on. I think a lot of those concerns have largely been addressed. We know that these myrianema infestations are only a problem at certain times of the year. So it's typically early summer, for example, in spring when the ulva starts to grow more rapidly. What happens is it strips out a lot of the nutrients, it causes stress, and then you get more infestations. And this can largely be mitigated by adding fertilizers into your tanks. If you're growing them in these integrated systems with effluent water, we've actually observed very, very little problems with myrianema in those systems. There was also some concerns around grazing back when the industry started, but this is again something that we haven't observed at all actually within these integrated systems. Threat that still largely applies even today after a lot of the research that we've done is the fear of spreading diseases. But as I've mentioned, we have done a lot of work. A lot of that recent research has now been published. In fact, what we are finding now that some farms that were reluctant are actually starting to adopt these technologies. However, amongst all of these threats that were identified back then, there were some new threats that have also been identified within recent times, and I'll quickly touch on those. One of them is threats around harmful algal bloom development. As I mentioned yesterday, this is something that is not only increasing in frequency in South Africa, but it is something that has increased in frequency globally. And in 2017, we had a major harmful algal bloom event along the coastline of South Africa, close to where the bulk of our large commercial abalone farms are. And you can clearly see that bloom, the adjacent coastline. And that actually caused a loss of more than 270 tons of abalone across 3 of these large commercial farms, and actually almost ended up closure of those farms. But as I've just told you, if you're practicing integrated aquaculture, you can actually isolate your farm and protect your farm from these harmful algal bloom events. So integration in this case is a huge benefit for farmers that are able to do so. Another threat that I think we've all realized in recent years was the impacts of large pandemics like COVID-19. And this had a significant impact on the industry, disrupting supply chains, increasing input costs on the farms. Farms couldn't export. They had huge problems with stocking densities on farms. So this all had a very detrimental impact on the industry, and very closely linked to that was the impact that all of that had on the markets, not only at the time, but long-term impacts. And together with what happened back in COVID and more recent changes that we've seen in markets, particularly in Asian markets, where we send the bulk of our products, that has also had sort of an adverse impact on the sale of abalone, and of course would also have an adverse impact on the production of ulva within these paddle systems. So this is the last slide. Sorry for taking so long. So just to wrap things up, just finally looking at some of the opportunities associated with ulva grown in land-based systems, looking at what was identified back in 2009 and what was identified to date. And we'll see that many of the opportunities on this slide are things that still apply today. So, for example, we've seen a significant expansion in the abalone industry since 2007. We've also seen a lot of new uses for UFR on these abalone farms. In particular, its use as a supplementary feed, which has huge benefits, as I've just demonstrated. But also, more recently, there is a lot of work going into the inclusion of dried UFR or components of UFR into formulated feeds, not only for abalone, but also for a variety of other species like sea urchins. And the next presentation will talk about that. That is obviously with including seaweeds as a supplementary feed that obviously has an impact on fish meal and fish oil use within seaweeds. So you're seeing a reduction in that. We're also slowly starting to see an increase in further expansion of these integrated systems. So we don't really have integrated abalone, fish, and ulva systems, but we are looking at a variety of other species for integration. So sea urchins, which I've just mentioned, and you'll see that in the next talk. We're looking at 2 different species that can be integrated in systems very similar to the abalone farming systems, and in fact also integrated abalone, ulva, and seaweed systems for some of our temperate water species. And something else which can also be seen as a strength and an opportunity is the support that we receive from government, which is now in recent years promoting more integration and particularly IMTA. So for me, that is definitely a benefit. Right, and that brings me to the end of my presentation. And yeah, happy to answer questions in the question session.
Merci beaucoup, Brett, pour cette belle présentation sur l'association de l'élevage d'algues d'ulva dans les systèmes d'hormones de recirculation partielle. Alors, très rapidement, on va passer à la deuxième présentation, celle de Maricia, qui a été préenregistrée, qui porte sur l'aquaculture des oursins. Donc, Maria, si la présentation est prête, on peut démarrer. OK.
Hi everyone, my name is Marissa Brunkal and I'm a production scientist with the Department of Forestry, Fisheries and the Environment in South Africa. In terms of my background, I've been working on IMTA systems with abalone and seaweed as well as with sea urchins for the last couple of years, together with the team that has joined me there in Senegal. I also have a background in genetics, where my recent work has been focusing on assessing the microbial communities associated with farm species and systems. But today I'm going to tell you a little bit more about sea urchin aquaculture, also known as echinoculture, starting with some general background, looking at the production cycle, and then I'll show you some examples of species that we have been developing technologies for in South Africa. So see. I think so, because you said that the subtitles in French were not good.
We can try it.
Yes. No. That's all that. That's fine. Oh, yeah. Thank you, sir. Screen. Okay. So, yeah. Very patient. It's not. Huh? Sambil bicara.
Hi everyone, my name is Marissa Brunkel and I'm a production scientist with the
Okay. Okay. Let's see you done.
Hi everyone, my name is Marissa Brunkal and I'm a production scientist with the Department of Forestry, Fisheries and the Environment in South Africa. In terms of my background, I've been working on IMK systems with abalone and seaweed as well as with sea urchins for the last couple of years, together with the team that has joined me there in Senegal.
Usman,
I
est-ce que vous
also
pouvez
have a
nous aider?
background in
C'est pas clair là,
genetics, where
l'écriture.
my recent work has been focusing on assessing the microbial communities associated with farm species and systems. But today I'm going to tell you a little bit more about sea urchin aquaculture, also known as echinoculture, starting with some general background, looking at the production cycle, and then I'll show you some examples of species that we have been developing technologies for in South Africa. So sea urchin gonads, also known as uni, which is what you can see on the slide here, is considered a culinary delicacy in Europe and in Asia, and as such, it has a high market value of, for example, $200 per kilogram on the Tokyo markets, and 80% of global sea urchin products, both produced in aquaculture as well as wild collected, are consumed in Japan, so this would be a key export target market. So globally, there are these two general schools of thought when it comes to sea urchin aquaculture. So sea urchins have been heavily overexploited in some countries, having a severe impact on natural populations, which then has subsequent effects as sea urchins are known to play important ecosystem roles in nature. But this decline in natural populations increases demand for sea urchin products from elsewhere to be taken advantage of if we can produce sea urchins on land by breeding them and closing the life cycle. In other countries, sea urchins are overpopulated and are decimating the kelp or seaweed populations, resulting in what is called urchin barrens. But this has led to the collection of urchins from these locations that would likely not have developed gonads as a result of not having access to food anymore. They are then taken to holding facilities and fed appropriate diets, quite often a formulated feed to encourage gonad development, but can also be fed seaweed, and then those are used for market to get a high price. And then in removing those urchins from the urchin barrens, the seaweed populations are given a chance to recover. In South Africa, and I believe also in Senegal, we really have neither of these scenarios, but we can firstly farm urchins to avoid future overexploitation, but also to diversify the aquaculture sector, which, as you've probably heard in this course, in South Africa is largely dominated by abalone. In terms of sea urchin markets, there are 20 common commercial species, but no reason why other species can't also be considered. This is especially true given the global demand with the reduction in global landings over the last 20 years, which is what you can see over here on the slide. And the price you get for urchins is heavily dependent on the product quality, as is the case for most farm produce, where you would get higher prices for live animals with bright orange or yellow gonads that are at a specific reproductive stage where they aren't yet producing gametes, as that would affect the firmness of the product. And then you get lower prices for gonads that have been processed by canning or gonads that are pale in color. So to talk about sea urchin aquaculture, we do need to briefly talk about their anatomy. So in the first image here, you can see their mouthpiece, which is what they would use to shred the seaweeds. And then you can see that they are organized pentaradially, which just means that they have 5 segments that are symmetrical. And at the bottom, you can see this internal cavity where they would have 5 gonads growing on the sides of the shells. And if you had to cut those animals open, this is what it would look like, which can be served like this for sushi or would be taken out of the shell. So to produce them in aquaculture, it does become quite important to close the life cycle, as this gives you more control over product quality, as specific feeds can be used during specific developmental stages to meet those nutrient demands, to promote growth and good gonad quality in the end. So in nature, sea urchin life cycles are characterized by males and females releasing egg and sperm into the water column as they are broadcast spawners. And then once the eggs have been fertilized, they develop into these prism-shaped larvae in around 24 hours. Thereafter, they stay as free-swimming larvae in the water column where they feed on microalgae. And here it is important to note that the larval phase is generally quite long and very sensitive. And while this acts as a challenge for producing them in aquaculture settings, through research we have been able to develop technologies to make this possible. And then once they are large enough and their settlement cues are present, they metamorphose by essentially folding in on themselves and enter the benthic phase of their life cycle. So based on the life cycle, land-based sea urchin aquaculture is also separated into two phases: the hatchery phase, the grow-out phase in a land-based facility. And the length of these phases really are species dependent, but this here is an example on the collector sea urchin Drypnestes gratilla. I am going to go into more detail on each of these steps, but briefly, sea urchin production would start with broodstock collection and conditioning, where there are fed specific feeds to encourage gamete production, and this is then followed by spawning, fertilization, and larval rearing. Thereafter, we have the settlement phase, and once they are little urchins, they are used to stock grow-out systems. And then the urchins go through a somatic growth phase, where their exoskeleton or shell grows for a couple of months to make space for those gonads. And then this is followed by gonad enhancement, where the gonads develop in that internal cavity until they are large enough for market. So now going into a bit more detail on each of these phases, one of the first things to consider is where broodstock are collected from, which will of course depend on the species of interest's natural distribution, but the genetic diversity of those natural populations should also be considered when possible, as this is what will be introduced into the cultured population. And particularly for broadcast spawning animals like sea urchins, this is quite important, as the diversity there can very drastically be reduced. So just as an example here, if we consider all these different colors, the genetic diversity that is present in the natural population, when there is a founder event which is caused by very few individuals from the natural population contributing genetically to the new cultured population, this can result in a drastic reduction in the amount of diversity that is present. And this reduced gene pool means that the cultured animals could have poor responses to stresses, poor responses to selection for specific traits, increased disease susceptibility, all of which could in the end result in poor production output from an aquaculture perspective. But we have also found that of each male with each female can avoid this founder effect. It shows for water as well. So everything that I see a progression of steps for different species. So once broodstock have been obtained, they need to be conditioned by feeding diets that encourage maternal investment of nutrients in eggs, And we know that this is best done by feeding them a varied diet so that they have access to a wider range of nutrients, which will in turn result in improved larval performance. Once the broodstock have been conditioned, spawning is induced by placing the urchin on a flask upside down, as the pores from which the gametes are released are at the top of the urchin, and that flask is filled with seawater to collect the eggs and sperm. The urchins are injected with a potassium chloride solution to induce the spawning, and then the eggs and sperm from individual animals are collected. Here you can see the females in the first row and males in the around the egg. From here, the eggs are placed into hatching troughs filled with seawater that has been taken to the species of interest's optimal temperature. And once they are hatched, the larvae are raised in containers filled with seawater, where the larvae are supplied with slow aeration and are fed with microalgae. This also means that while you're feeding or raising sea urchin larvae, microalgae need to be maintained and grown up into larger volumes. And here you can see 3 species that are commonly used as they are good for sea urchin larval development, and that's Rhodomonas, Catosphorus, and Isochrysis. And we generally feed the larvae at 10,000 microalgal cells per larva, which means that the number of larvae and the amount of microalgae needs to be continuously quantified by counting cells under a microscope to know what volume to feed a tank of larvae. In these larval systems, water quality is one of the key parameters to monitor, particularly because of the microalgae that gets added as feed. So it can be run as a static system where full water exchanges are done by fully draining tanks and collecting the larvae and then placing them into a tank with clean seawater, or water can slowly be trickled into a system so that it becomes a flow-through system where water is slowly exchanged over time. And then if all goes well, these images here below show you how the larvae should develop over time from a 2-armed stage to a 8-armed stage over about 35 days. And here you can see the stomach, and we know that they are feeding. As you can see, that slight yellow tinge, which shows you that there is microalgae present in the stomach. So thereafter, larvae are monitored to see if they are ready to be moved to a settlement substrate. And one of the things we look for is the structure here at the top, as that is what they will end up using to attach to a substrate. And settlement readiness is also evaluated by looking at this black structure known as the rudiment. And we know that they are ready for settlement when this structure is larger than the stomach. And then the presence of tube feed, which you can start seeing here in the second image and very clearly in the third image, is of course another indicator that they are busy turning into juvenile sea urchins. What I like to use is Ulvella lens, a crustose microalgae, which is this green mat you can see over here, as this is quite a successful settlement substrate for sea urchins. And then shredded ulva, the green seaweed, can also be added to induce a settlement until you have these little urchins crawling around everywhere. So the juvenile sea urchins, who are ferocious feeders, are then fed ulva until they reach around 10 millimeters in size. And then they are moved to a grow-out system where they are generally held in these baskets so that the fecal matter and uneaten feed can fall through the mesh, firstly making it easy for easy cleaning, as the baskets can be removed to give the tanks a thorough clean. And then, because sea urchins tend to congregate on the sides of baskets, one aspect that we need to consider in the grow-out phase for sea urchins is basket design, as it could impact spine loss, which means they need to invest energy into repairing those structures rather than into growth. It could also affect their consumption, so how they feed, and could affect the extent of damage when they are lifted out of the baskets for tank routine cleaning, as they would drop off to the bottom. And then depending on what height they are dropping from, this could affect how damaged they get. So when one of our PhD students looked into this, he found that while basket depth had no effect on spine loss, the type of feed did. So for example, you can see here that kelp-fed animals had significantly higher spine loss when compared to formulated feed and ulva-fed animals. And we think that this is mostly attributed to the breakage of spines when removing uneaten kelp from the baskets during cleaning. And then when looking at the impact of basket removal during cleaning, it was found that when baskets are removed for a longer period of time, higher spine loss was observed. And this was particularly true for deeper baskets, and this is likely as a result of the urchins losing grip on the sides of the baskets over time if they aren't submerged in the water. So when looking at how basket depth affects consumption of feed, we found that sea urchins are pretty lazy creatures, so they do prefer shallow baskets where they don't have to do much effort to get to their food. So here you can see that there was higher consumption for both formulated feed and ulva in a shallower basket when compared to the deeper ones. And then another important consideration is stocking density, which can be calculated either based on the animal weight and the available surface area in the basket, or it could be based on the amount of space that each urchin would take up on that internal surface area. When we tested low, medium, and high stocking densities, we found that lower stocking densities result in faster growth rates, but that higher stocking densities resulted in greater total yield at the end of the production period. So to maximize overall yields, higher stocking densities can feasibly be used. So the length of this grow-out period is dependent on species, the system, and the types of diets that are used. And that is why it's important to develop feeds and optimal feeding regimes when starting aquaculture of a new species, especially for sea urchins, as they have those two phases of somatic growth and gonad enhancement, during which they would have different dietary requirements for optimal growth. And then they are considered ready for market when the ratio of their gonad weight to their body weight, also known as the gonad somatic index, as a collaboration between the DFFE and the University of Cape Town, have been working on developing aquaculture technology for the collector sea urchin and the Cape sea urchin, as these have been identified as candidate aquaculture species. The collector sea urchin occurs on the warmer east coast of South Africa. It's an internationally traded species. It has a fast growth rate of about a year to market size and produces these bright orange gonads, which is what you can see over here. And it can be produced in IMTA systems with ulva, and I'll show you some examples of that in the next couple of slides. The Cape sea urchin, on the other hand, they are very abundant along the South African coastline and have the potential to supply a local market, but could also be traded internationally if product acceptance is high enough. And as they produce bright orange or red gonads, this may be possible. The species also has potential for integration with ulva or with juvenile abalone, but I'll get into this a little bit later in the lecture. So focusing in a bit first on the collector sea urchin, extensive research has been conducted on the species, where our research group has developed feeding regimes, as well as a formulated feed that contains the green seaweed ulva, which has been shown to improve gonad quality, and it's this feed that you can see in this image over here. And then we have also conducted population genetic studies, developed breeding protocols, hatchery techniques, and grow-out technology. And then we have also conducted microbiome and disease studies and developed software to measure urchins efficiently, especially at a farm scale. And then the next step was to move from research scale to commercial scale, which can act as quite a challenge for any new aquaculture venture. So to do this, we reared larvae to stock a pilot commercial system with 30,000 sea urchins, using all the methods that I've been discussing throughout this lecture. And if you are keen to see more details on these protocols, feel free to check out the production manual that we put together during the course of a EU-funded project called the Stroll, and this production manual is freely available online on this website displayed on the screen. And then once the urchins are large enough, they were stocked into these baskets at a stocking density of around 8 kgs per meter squared. And this system specifically was built in this tunnel that you see here. This was done to conserve heat, as the species prefers warmer conditions of around 25 degrees Celsius, and we had to run a heat pump, as this farm is on a stretch of our coastline that is a little bit cooler, so not necessarily the optimal spot for the species. But inside this tunnel, we had a system that looked something like this. And you may have seen something similar shown for abalone during this training course. But we stuck the urchins into baskets that were in tanks that were linked to a ulva paddle raceway. And this was, like I said, modeled on that abalone ulva system. The urchin effluent goes through a drum filter to remove the particulate matter, and then the dissolved nutrients flow through this ulva raceway, where it acts as a nutrient source for the seaweed. And then after running through the raceway, it is mixed with 10% fresh seawater, and then this is returned to the sea urchins. So here we had a really high recirculation rate of around 90%, but again, this was mostly done to conserve heat. So there are several aspects that need to be considered when implementing a new INTA process, particularly when it's moving towards a commercial scale. First of all, the cycling of nutrients. You also need to consider the performance of the seaweed, so of the ulva, and in this case we needed to think about temperature tolerance, as this ulva strain is generally grown at 16 degrees Celsius with the abalone, and we had to see whether it would tolerate 25 degrees Celsius. And then we also need to consider basket design together with stocking densities. And then, of course, sea urchin performance needed to be assessed in this pilot commercial INTA system. We implemented 2 feeding strategies to assess what would work best for commercial production. In one of the feeding strategies, we had the animals fed on fresh ulva that was produced in the system. And this was done for 5 months, so for that somatic growth phase of production. And then we swapped over to a formulated feed for the remainder of the production period to bulk up the gonads. And we did this across 2 formulated feeds. And then our other feeding strategy was to have the urchins on a mixed diet of ulva and formulated feed throughout the whole production cycle. And for this, we used 3 different formulated feeds. But we found limited growth differences across the diets. What we can see is differences in gonad development between the different diets or feeding regimes, showing us here in yellow and in green that it is best to feed them ulva for the somatic growth phase and a formulated feed for the gonad enhancement phase. This trial also showed us that the species can be produced at a commercial scale and be ready for market in about a year, which is quite impressive if you consider that abalone takes 3 to 5 years to get to that point. When we looked at gonad quality, the feedback we received from restaurants was quite positive in terms of the flavor profile and other sensory qualities like texture, but the diets did not all perform well when we look at gonad color, with the last 3 columns that you can see here being a little bit more marketable than the first ones that are more pale. And as color is a key determinant of marketability, this showed us that we do need to further refine the feeds for optimal production. And sea urchins, color is thought to be influenced by carotenoids, which are naturally occurring red, orange, or yellow pigments. They are usually of plant origin. And in the case of this work, beta-carotene is being obtained from the ulva, as well as from the ulva that has been incorporated into the formulated feeds. And besides all the other benefits that the animals are getting from the incorporation of these pigments in their tissues, we can still play around with its inclusion in sea urchin diets for improved gonad coloration. So moving on now to the second focal species, which is the Cape sea urchin. We started working on this species for a completely different reason. We wanted to see if we could take advantage of the known relationship between juvenile abalone and the Cape urchin along the South African coastline. In nature, the abalone are often found under urchins over 90% of the time, and this is likely for improved food supply, shelter, and protection. But we also hypothesized that there could be a transfer of microorganisms from the urchins to the abalone that could benefit the digestion while their digestive systems are still immature and developing in that juvenile abalone phase. And then because these two species have quite similar temperature preferences, this was represented an opportunity for INTA as well. So to test this, an 8-week experiment was conducted in a commercial abalone hatchery where we had 2 sets of 3 tanks, where each tank was stocked with 25,000 juvenile abalone that were just moved off of their settlement site, the mixed diet, and this formed our control. And the other set of tanks was fed this mixed diet, but also supplemented with sea urchin fecal matter collected from 310 sea urchins per tank. And during this time, the sea urchins were only fed ulva and were held in baskets in tanks separate from the abalone. So you may wonder why not just put them together in a single tank, but when we tried this, and we tried many different configurations in preliminary trials, we found that the animal husbandry practices needed to be simplified to streamline incorporating this in a commercial farm in a realistic way. So this is where it becomes important to work side by side with the industry. And we found that the juvenile abalone that received the fecal matter supplement had improved growth, and we also found that beneficial microorganisms that are capable of synthesizing digestive enzymes and fatty acids and are known to play key roles in the assimilation of nutrients were transferred from the urchins to the abalone. And the abalone also had access to protein in the early hatchery phases where they would normally not have access to formulated feed yet. So this presented a benefit to the juvenile abalone farming practices. But this led us to ask whether the Cape sea urchin couldn't also be a new aquaculture species and another product from this integration. So at that point, very little was known about the Cape sea urchin growth, gonad quality, and subsequent marketability under aquaculture conditions. So a master's student of ours ran a 6-month feeding trial with wild-collected urchins. We had 2 temperature treatments to see if slightly warmer water could result in faster growth. And we tested 4 diets using our prior knowledge on the technologies developed for the collector sea urchins. So interestingly, we found that the seaweed kelp wasn't a suitable feed for the species, which was somewhat surprising given the high abundance of Cape sea urchins in the South African kelp forests. But this decline here is, or decline in survival, is depicted here in yellow across both temperature treatments. And so this led us to exclude this dietary treatment from the rest of the experiment. And then we found that a mixed diet of ulva and formulated feed resulted in the greatest sea urchin weight gain across both temperatures, which is what you can see over here in blue. And not only does this inform us on what to feed them, but we could also see that when they are cultivated at 16 degrees without needing to warm the water, it makes them quite compatible with abalone farms in the Western Cape, as abalone are also farmed at that temperature. So this species produced bright orange and red gonads when fed ulva or a mixed diet, and unmarketable gonads when fed a formulated feed. But what I want to highlight here is how the males produced pale or unmarketable gonads. So it was clear that there were some sex-specific differences in gonad quality for Cape sea urchins, which is something we haven't seen in the collector sea urchin before. So we are still in early stages of developing this aquaculture species, but we have recently developed a method to identify whether they are male or female without sacrificing the animals by looking at the gonadal pore structures. But looking into future work, we need to develop optimal feeding regimes for the species. And we also need to start considering what other uses we could have for gonads from the males, like incorporation into other seafood products or fortified food. And then looking at the next steps for sea urchin aquaculture in South Africa, commercial production of the collector sea urchin is possible, but would be better on a stretch of coastline with warmer waters to remove that cost of having to run a heat pump, which shouldn't be a problem in Senegal. We also need to further develop the formulated feeds that incorporate ulva as a source of carotenoids, as this is needed to promote good gonad quality and color in particular, and this is applicable across both species. And then we also need to start looking into the nutrients that are present in the particulate matter, as this could be recaptured for use by another species rather than being passed through a dump filter and disposed of. And we have been looking a little bit into whether this could be used to farm bloodworms and whether they could make use of sea urchin fecal matter as a food source. And then for the Cape sea urchin, we are looking to adapt the collector urchin technology to meet the Cape sea urchin requirements. And we are also aiming for full life cycle grower to improve our understanding of their growth rates through various developmental stages, and to assess the impact of different feeding regimes on growth, gonad quality, and market acceptance. So sea urchin aquaculture, as with any farming practice, does not come without challenges. The first being that post-harvesting processing and transport of sea urchins to restaurants or overseas markets needs further development, as products can quickly deteriorate if this is not done well. Next, the familiarity and acceptability of sea urchin products food challenge, as it is not necessarily that, or not necessarily something that we are locally very accustomed to. Can be done by adapting our abalone farming technology for sea urchins, which means that may not be needed. And then we need access to and in in the South African abalone industry. Well, and the rock boring urchin. Neither of these are commercially farmed elsewhere due to their small body sizes and are commercially farmed elsewhere due to their small body sizes and the slow growth that would normally take around 3 years to market size, in some cases even 5 years. And this, together with the low ratio of gonad weight to the overall body weight, or the GSI, which for this species gets around 8%, which is substantially lower than the market standard of 20%. In contrast, the long-spined sea urchin can get up to 12 centimeters in diameter, but is generally not commercially farmed because of their long spines that make them really impractical to handle. And then they also have quite small gonad sizes relative to their large body size, with maximum GSIs of around 7%. And then the last species I want to highlight is one that I believe has aquaculture potential, and it is the sea egg, or white urchin, or tropical urchin, which is very closely related to the collector urchin that I've been discussing in this lecture. The species grows fast, can be grown to market size in about a year in full life cycle grow out, or can undergo gonad enhancement in just 4 to 8 weeks. And it reaches that market standard of 20% GSI. So I do think it holds some promise as a candidate for aquaculture. And with that, I would just like to thank you for your time, and you're more than welcome to contact me on email if you have any specific questions. Otherwise, I'm sure my colleagues that are there with you will be able to address those too. Thank you very much.
Merci beaucoup. On va enchaîner très rapidement pour les discussions, concernant les deux présentations. 2 à 3 questions maximum avant de prendre la pause café. Il y en a 2 à 3 questions rapidement. Oui, il y a Ndiaga Ndiaye. Et qui d'autre? Madiké. Un troisième intervenant, s'il y en a. Ok, donc on se limite à ces 2. Ndiaga après Madiké. Hein?
Voilà. Merci Mafal. Merci Monsieur Lum, de m'avoir donné la parole. Et également, merci à Kevin pour la clarté de la présentation. Moi, j'ai deux petites questions par rapport aux bactéries que vous avez mentionnées lors des présentations. Par exemple, dans vos résultats, vous avez montré qu'il peut exister plusieurs bactéries dans un système d'élevage. Maintenant, j'aimerais savoir, est-ce que vous avez pu caractériser ces bactéries afin de connaître celles qui sont néfastes pour le système et celles qui sont bonnes? La deuxième question, vous avez également souligné qu'il y a des bactéries qui peuvent intervenir dans la morphogénétique. Maintenant, est-ce qu'on peut avoir plus de détails sur ce point-là? Comment cela se passe réellement? Merci.
Can I go ahead and answer that one? Yeah, thank you very much. That was a very good question. So I think the first one that you were interested in was just the bacteria within the system, wanting to know whether we had characterized the bacteria within that system. The answer to that question is yes. So I didn't go into detail around the analytical tools that we used to do that, but we used a next generation sequencing approach. So essentially what we did is from each of those samples, we extracted all the DNA and then we amplified up all the bacteria that occur within that system. And using a bioinformatics approach, we were able to characterize all the microorganisms within that system down to a genus level. So we typically, for those analytical type tools and using that approach, you can go down to a species level, but it's not very meaningful because you're looking at very short fragments. So you've got to be very careful with the interpretation of the information that you get if you do that. So genus level is typically how far you go. But yeah, so we do have comprehensive data on that for each of the different compartments. And we have actually published some of that from a different system. We've got a manuscript that is about to come out from one of our systems at Biffeljaks, where we've been doing a lot of our recent studies. So as soon as that comes out, I would be happy to share that. And what I will do is I'll share the other publications with Mbaye, and he can share that with you as well. And then regarding, I think the second question was around the bacteria that are associated with ulva, looking at the morphogenesis and the growth. There's a lot of information available on that. So we did identify things like Maribacter, which is one of the species that's involved in that process. And there's a second one, I cannot remember off the top of my head right now, but I'll also send you some more information on that. But these are symbiotic bacteria that you will find associated with the seaweed. They secrete certain compounds that are very important for that process to take place. And in their absence, that process will not be completed. So, like I said, there's a lot of data. We haven't ourselves intensively studied those mechanisms, but there are several groups in Europe that have done that. There's actually another big project which has also just come to an end, which was called COST, Ulva Wheat of the Sea. And there's an extensive report from that project as well, which I'm happy to share with you. And I think it's got a lot of literature and references in there, which will give you a lot of very detailed and intricate information around that whole symbiotic interaction between the ulva and the bacteria. So again, I will share those with Mbaye.
Madické.
Bonjour à tous.
Ah.
Donc, merci, monsieur Brett, pour votre présentation très intéressante. Donc, ma question est la suivante. Alors, quelles sont les conséquences que peuvent avoir le système d'intégration hormo-algues causé par les impacts négatifs, notamment sur l'environnement, ainsi que sur les produits dans le long terme? Merci.
Thank you very much. Again, an excellent question. was even though we have been doing a lot of research in this area, it doesn't mean that we have fully resolved everything. And it certainly doesn't mean that we've actually fully optimized the system. So in terms of negative interactions on the environment, it's actually quite a good one. I can't think of any major impacts at the moment besides the floor, the foot space or footprint that a farm could potentially have, because all of our farms are obviously land-based systems. They're obviously located very close to the coastline. They can obviously be located sometimes in very pristine areas. So I think one does need to be very careful about site selection and making sure that you don't have a negative impact on sensitive areas, like going into mangrove areas or areas that have sensitive vegetation. So I think that could be one of the negative impacts on the environment. And in terms of impacts on the product, that's actually an excellent question, because that is something that we're actually looking into a lot more at the moment. So in my talk, I mentioned we've been looking a lot at the physiochemical parameters of these systems, but that was really done just to get some baseline information that would enable us to interpret what we are seeing with regards to the microbiome within these systems, because obviously that variation that you're going to see is going to be directly dependent on some of those factors. But we actually need more information on that. And it's not actually a simple thing to do. In order to intensively monitor these systems, you require quite a lot of equipment that is actually quite expensive. And we've just gone through a process now, myself and Maria, to fund the procurement of some more equipment in order to support ongoing studies to get really detailed, continuous data of what's going on within these systems. And then what we're going to be doing is we're going to be trying to see whether we can further optimize that system. So we know there are benefits. Those benefits appear to be more beneficial at certain times of the day. Whereas in the evenings, for example, in an abalone system, some of those parameters are becoming a little bit detrimental. pH is quite low, oxygen drops, there's high nutrient levels. So we're looking at different configurations of running the systems, maybe looking at switching to either lower recirculation or full recirculation at certain times of the evening, so that we can mitigate against some of those things. But still get the full benefits out of the integrated system and the ULVA that we have within that system, because we all regard, I think, in our group as hugely beneficial to the health of the animals and the overall functionality of those systems. I hope that answers everything.
Ok, merci beaucoup. Donc, on prend un dernier intervenant pour cette partie. Professeur Lee, vous avez la parole.
Bonjour tout le monde, merci pour les brillantes présentations. Je vais donc poser une question en ce qui concerne l'élevage du sea urchin. La question est de savoir, est-ce que c'est uniquement les gonades qui sont consommées auprès de sea urchin? Si c'est le cas, est-ce qu'il ne faudrait pas seulement élever donc les femelles, d'autant plus qu'elles présentent des performances beaucoup plus importantes, et ou bien trouver un moyen de renverser ou phénotypiquement utiliser uniquement des urchins qui arrivent à développer donc des gonades beaucoup plus importantes. Autre chose, je vois aussi une coloration différente des deux gonades produites. Quelle substance ou bien produit est-il possible d'ajouter dans l'alimentation pour pouvoir renforcer cette coloration en fonction, par exemple, de la couleur qui est la plus préférée par les populations ou les consommateurs? Ce qui m'amène aussi à me poser la question, à savoir, sachant que les algues, les gracilaires en général sont très utilisées dans ces systèmes intégrés avec ces animaux, est-ce que, sachant que les algues coûtent très cher, est-ce que c'est un système qui sera rentable pour les producteurs moyens ou la grande masse des producteurs? Merci.
Great. Thank you very much, Professor Lee. Lots of questions, so I hope I've captured them all. If I forget anything, just let me know and I'll respond to you. Again, all very, very good questions, and I'm glad you've asked them. So I think the first one was regarding the gonads and the difference between the male and the female gonads, and whether we can do something in order to select female gonads which are better quality as opposed to the male gonads. That's is a really good question, and it is something that varies actually between different species. So in the tropical sea urchin that Marissa spoke about, Trypneustes gratilla, you do get a variation between the male and the females, but the extent of that difference is actually not that great compared to the other sea urchin, the temperate sea urchin species that she spoke about, Parachinus angulosus. And in that species, we actually find a very, very big difference in the quality of the male and the female gonad, with the female gonad being far more marketable than the male gonad. So there's different ways of getting around that. One thing that we had thought about at a stage was whether we could very early on in the production cycle differentiate between the males and the females. And in fact, we had a student that from France who did a short internship with us, and he actually developed a very, very nice key that we can use. So things like that have been done for other urchin species, and we validated that for our local species. And we're hoping that we can further develop that technology, that we could make it applicable on a commercial basis. And so using things like AI and other modeling techniques that can be integrated into grading on farms, which will enable us to differentiate those. So that is possible to early on differentiate. The other thing that one could do is to make dietary interventions to try and see whether you could further enhance the quality of the male gonads. So potentially develop sex-specific feeds. We haven't started on that yet, but that is something that we are going to be exploring pretty soon. And then I suppose one could also maybe think about using other tools to manipulate it, maybe triploidy or something like that. But again, that isn't something that we've actually got to at this point. But another thing that we've also been thinking about, particularly with Parachinus, is that if the male gonads are not marketable, what can we do with those products? And there's huge potential to do other things with them. So as Maria spoke about in her presentations, that we've been looking at the nutritional value of the products that are produced from these species. And gonads obviously are very, very rich in a variety of minerals and fatty acids, and they hold huge potential for being used. So, and vitamins, yeah. So they hold a lot of potential for other uses, potentially in other food-based products. So again, that's something else that we can explore in order to ensure that we're getting more value out of waste products, if you want to call it that, coming out of the production system. And regarding what is the main product? Yes, it's predominantly gonads coming out of it. At the end of the production cycle, in that animal, you can get a gonad somatic index as high as 25 up to 30% even. So when you open up that test, the shell of that animal, it is literally just full with gonad material. So that is predominantly what you're going to be looking at for market. There isn't really anything else besides the test itself. And Maria also mentioned yesterday that that's very rich in calcium and other minerals. So I think there's also something that one could do with that byproduct or waste product in the system if it's dried and used for other purposes. In terms of the coloration, I think the other question was what can we add in order to improve the marketability of that gonad and the coloration of that gonad. 100%. There is obviously, feeds are obviously very important from that perspective. And we've done a lot of work looking at seaweeds, for example. There's a lot of literature that shows that seaweeds are a better feed for these animals because they've got a lot of pigments in them, things like beta carotene, which is actually quite abundant in ulva, besides the fact that it's a green seaweed. It's got a lot of beta carotene. And that is actually the reason why, because it was so available on our abalone farms, why we looked at incorporating that either as a feed or as a feed supplement for these animals. Because our data has clearly shown that it has a significant impact on the coloration of those gonads. So what we typically do is we'll grow these animals out predominantly on ulva as a feed. So you get a very nice colored gonad initially during the production cycle. But those gonads are obviously quite small. And once the animal's test is large enough, we will then switch over onto a formulated feed that also contains a certain percentage of ulva in it so that we can maintain the color. But the high protein content of that formulated feed then allows that gonad to increase in size. So it then fills up that space within the test. So that makes the entire farming operation a little bit more profitable if you have access to large amounts of seaweeds. But of course, if you don't, we've also shown that these formulated feeds supplemented with these seaweeds is sufficient to produce a marketable, good colored gonad of sufficient size. So you can use that throughout the production cycle. And then I think the last question, if I understood correctly, was growing seaweeds in systems with this species, sea urchins, whether or not that is profitable or not. Again, like I said to you earlier on, I think it all very much depends on site, what the farmer has available to him. So my opinion is that if you do have the space and you are trying to grow this species, I would do it in a system that is integrated. I think the integration has a lot of benefits to the animal and to the system as a whole. As Marissa pointed out, it is a very like a high recirculation rates that we typically use within these systems, particularly for the tropical species. And we need to do that to maintain temperature. If you are doing that, you're going to need some form of biofiltration in that system. And we know that seaweeds are a very, very good biofilter within that system. So we ran our large commercial scale systems purely with ulva and cattle raceways as the main biofilter within that system. The only additional component we needed within that system was a drum filter to remove particulates before the waste water went into the ulva paddle ponds. So if you can run your system like that, then you've actually got a feed and it grows exceptionally well. We had super high growth rates of the ulva in actually a very small paddle pond that was more than enough to provide a feed for the animals in the system. It was very high in protein, so it actually was a very good feed, and it provided us with a very good feed for enhancing the coloration of that gonad. But as I've said, if you don't have that and you are in an area that is more suitable to the production of a tropical species, for example, then you can actually also just use a formulated feed, and you might not need to have a seaweed integrated in your system. So it's not essential. And then coming back to some of the colder or temperate water species, like Parachinus that we currently focusing a lot of attention on. For me, that species is an absolutely amazing species with huge potential for integration into our current abalone integrated systems, because you can literally just slot it in. The technology is exactly the same. You can just dump it into baskets if you've got baskets available in your system. And the only thing we need to do now is just make sure that we can get a consistently good product out of that animal.
Merci beaucoup, Brad, pour ces réponses apportées à ces questions concernant les deux présentations. Alors, pratiquement, il est 11h30. Donc, ce qu'on va faire, on va observer une pause de 15 minutes pour permettre à certains de pouvoir recharger leur batterie. Voilà, donc on reprend à 11h45. Hein? Voilà, allez, merci. 15 minutes. Hein?
Je voudrais faire une petite précision par rapport à la traduction que vous étiez en train de lire. Vous avez vu qu'il y avait le mot arachide de temps en temps? C'est pas arachide, c'est gonade. D'accord? Ok. Désolée pour ça.
Alors, je pense on peut redémarrer. Ouais, allez. C'est bon.
Bonjour à nouveau. Mon collègue, il est en train de distribuer une petite feuille avec une activité on fera après la pause la pause déjeuner, certains. Et, euh, et donc, je vous invite à la lire, voir de quoi il s'agit. Et en fait, ce qu'on vous va demander après, c'est de vous distribuer en 4 groupes selon vos affinités, comme vous voudrez. Mamie a compté, vous êtes autour des 40-45. Donc, ça va être des groupes de 10 personnes, 10-12. Ne, que l'activité ne fasse pas vous peur. C'est juste un exercice sympa pour vous d'essayer de penser comment, si vous êtes dans une situation de développer une ferme aquacole, qu'est-ce que vous ferez en fonction de votre intuition, mais aussi en fonction de tout ce on vous aura parlé pendant ces 2 jours-là. Donc, profitez de ces moments pour réfléchir à ça. Et puis, on vous donnera peut-être 45 minutes pour réfléchir ensemble à chaque groupe. Et vous allez mettre un petit schéma. On va vous donner, on va placer une espèce de grosse feuille sur le mur et on va vous laisser créer ces petits dessins des fermes aquacoles et vous aurez dessiné quelqu'un ou une ou 2 personnes ou si vous voulez 10, mais bon, c'est mieux que vous dessinez quelqu'un qui explique un peu ce que vous avez fait. Puis, avec ça, on verra 4 modèles de fermes aquacoles que vous auriez dessinés et on va discuter sur ce que vous avez fait, les choses qu'on vous demande dans la feuille et on va discuter de tout ça ensemble. Et bon, pour nous, on a fait ça souvent dans nos cours de formation et ça vous met dans une position pratique qui est très intéressante puisque la théorique, à fin de compte, voilà, c'est de l'appliquer à un moment donné. Donc voilà, c'est tout. On vous dira quand on commence cette petite réflexion. Merci.
Ok, merci beaucoup Maria pour ces précisions. Alors, on va continuer les présentations. Maintenant, nous allons donner la parole à Kevin pour faire sa présentation d'aujourd'hui. Kevin, vous avez la parole.
Merci beaucoup. Interpreters, can you hear me okay? Great. All right, so today I'm going to be talking a little bit of a continuation on where we left off yesterday. And I've entitled this talk after a quote from the famous movie The Godfather from 1972. And that says, closer to the mic. The quotation from the movie is, keep your friends close, but your enemies closer. And I thought that's quite appropriate for the topic of today's lecture, which is about monitoring pathogens and disease risks in aquaculture through observation, extension services, and surveillance. As we learned from the syndemic pathway I presented yesterday, Aquaculture systems are biological production systems where hosts, pathogen, and the environment interact continuously. Disease occurs when this balance is interrupted. Modern aquaculture faces several drivers that increase the risk of disease outbreaks. These include intensification of production, movement of live fish and fingerlings, environmental stresses, and obviously climatic variability. As I said, the title, Keep Your Friends Close, but Your Enemy Closer. In an aquaculture context, this would mean understanding the biology of the pathogens that we are facing, continuously monitoring your fish and your fish health in the system, or oysters, or whatever aquatic organism you're culturing. Detecting disease risks before outbreaks occur. And as I said yesterday, effective disease management relies on early detection, which in turn relies on monitoring and surveillance. Human activities such as feeding practices, stocking density, and water management also influence disease outcomes, as we heard yesterday. Therefore, disease monitoring must be considered within the entire farming system, not only the pathogen. Diagnostics help determine the cause of the nature of the disease through the examination of clinical signs and laboratory tests. These form the scientific basis for the disease control strategies. Monitoring and surveillance are terms often used in aquaculture in this context. They are very similar. However, monitoring refers to routine observation of fish health and environmental conditions. Typical indicators on your farm will include mortality rates. Maybe your fish aren't feeding like they usually do. If they're going off their food, obviously they're growing slower. You might be measuring environmental parameters in which your water quality parameters are not right. Either your oxygen is not high enough or you've got excess ammonia in the water. And then obviously handling your fish during grading or just physically observing them for lesions is also a good indicator. Monitoring is primarily conducted by the farmers, farm technicians, and extension officers. So these are the areas in which you can implement surveillance and observation and early detection in your own system. Surveillance, on the other hand, represents a systematic collection and analysis of health data across multiple farms or regions. Surveillance is typically used to detect disease outbreaks, to track the spread of pathogens, as you can see on that second item, to inform policy and biosecurity measures, and ultimately to support trade. Disease surveillance serves different purposes along the continuum of risk management. And these purposes include early disease detection. Usually, as we've spoken in the previous slide, this could be erratic swimming, reduced feeding, and it allows rapid interventions before major losses occur. And that's really important of early detection, is that it allows you to do something about the situation before you lose more animals or before the disease spreads into the wild or to your neighbor's farm and the losses become greater. Routine health monitoring is the second purpose that we use surveillance. routine health monitoring, that is looking at seasonal patterns of disease occurrence, pathogen prevalence, whether there's a change in the amount of animals that are infected in the system. Are there environmental risk factors that we need to consider? And all of these together help improve farm management or help inform the management procedures and extension services. Something's happening here. There's a Zoom opening. Sorry.
That internet connection.
No. So I could just carry it.
No, because it's so. Check another one.
Merci, Maria. All right, as I was saying, there are a few reasons why we do conduct surveillance, early disease detection, as I've said, routine health monitoring. The other reason we'll do targeted surveillance is when there's an outbreak and we're doing an outbreak investigation. Remember the scenario we created yesterday where you were the investigator, and that would be what you would be doing. You'd be doing a surveillance exercise to support the investigation of what's causing the disease, to try and get confirmatory diagnosis. And confirmatory diagnosis allows for effective treatment strategies. If you've had an outbreak and you're doing an investigation, clearly there's been a break in your biosecurity system. So the targeted surveillance helps inform your biosecurity revisions, and obviously will inform movement restrictions. You're not going to move animals from an infected area to an uninfected area. The fourth reason for doing surveillance is for target surveillance, and target surveillance focuses on the specific high-risk pathogens. There may be pathogens of local importance or at a country level. They generally have some regulatory controls, and the most common examples of these diseases would be those diseases which have trade implications and are listed by the World Organization for Animal Health. So it is applied when a disease emerges nearby to inform containment or zoning. So if you have an outbreak of one of these diseases, you'll do targeted surveillance of the infected farm and the surrounding farms, and you'll spread that out to eventually see a zone, a geographic area that represents the infected farms. You would look at a zone outside of that where you have no disease detection, but that will be regarded as a buffer zone. And then outside of that, you'll have another zone which has also no disease detections. And those zones, the free zones, will still be free to trade in the aquaculture products. The last one is for health certification, which is pretty much what we do in South Africa for our abalone. We do screen healthy animals using targeted tests to confirm that they are free of these trade limiting diseases or diseases that are listed by the World Organization for Animal Health. International trade or transboundary trade of aquatic animal products or any animals or animal products will require some kind of health certification. And that is the backing for the fifth reason for doing targeted surveillance. technical issues. It's not advancing the slide. Ah, yeah, I'll use that one. Yeah. Merci, Maria. All right, so for surveillance systems, we have two types of surveillance when we're doing. So we know what the reasons are for doing surveillance, but there are various ways of conducting surveillance activities. The first one is passive surveillance. Passive surveillance is a term you should be or you will become familiar with. It relies on voluntary reporting of disease events. Where do you get these information from? So these are farmers, extension officers, hatchery, community networks that will report disease outcomes or to a competent authority or a veterinary authority. So the information is voluntary. It's coming from people on the ground, and it's information that is fed to the regulators or the veterinary health control boards. It has advantages. Passive surveillance has huge advantages, is it generally costs very little. The burden of evidence for passive surveillance is based on observations. So there's no expensive testing. It is done by people on site, so you do not need to travel. So it's really cost effective, and you can cover a large geographic area at the same time. It does, however, have some limitations. The limitations are related to underreporting. So the passive surveillance system only works when you have everyone reporting in an area. If other people or some people are not reporting, then you have breaks in the data set, and that is obviously a weakness. And as I said, it's based on observation, and generally the farm workers or the people handling the animals are constantly observing these animals. So it's extremely sensitive because the animals are under constant surveillance. However, the specificity is very low because you're observing clinical signs which are not necessarily specific to a certain condition and may be general. So the diagnostic confirmation level is very low in passive surveillance. Despite its limitations, passive surveillance is the foundation of aquatic animal health systems in many developing countries. As opposed to passive surveillance, active surveillance involves planned data collection. Examples include scheduled farm inspections. You can see on the top left one of our inspectors on an abalone farm. You have fish sampling programs which are planned. You have water quality monitoring, and this can either be continuous through instruments that are left in site, or you have a schedule of times for water quality monitoring and laboratory testing. As you can imagine, active surveillance provides higher quality epidemiological data. But on the other side, it does require a greater financial and technical resources in order to get that level of resolution in your data. We've spoken about what needs to be done in terms of surveillance. The next component is how is it done? Ultimately, a level of observation or diagnosis is required to inform your surveillance programs. And the FAO, along with the Food and Agriculture Organization of the United Nations, FAO, have developed a 3-level diagnostic procedure. So level 1 is the diagnostics which typically occur on the farm, and they rely on your observational skills. As I've said many times before, what you're looking for is fish behavior. Are the fish swimming in the group, or are they swimming separately? You can see that one fish swimming off to the side on the image and all the fish gathered together. There are external clinical signs. Fish can be darker in color than the rest, or fish can be lighter in color. They can have physical lesions. They can have bloated stomachs, bulging eyes. All of these are external clinical signs. Environmental conditions. Has something changed? Has the oxygen dropped? Has there been a sudden spike in temperature? Is it following a rainstorm, a sudden inundation? Has salinity increased or decreased? Those are all environmental conditions which you can observe at the farm level. And obviously there's a history. So when you're doing these things, particularly when you're looking at mortality data or the number of animals that have died, you look at these over time and you see whether there's been a change over time. And if you're monitoring a lot of these things and you keeping the records, as we mentioned in yesterday's talk, you can have a historical view and you can see when suddenly something has changed in your observations, and that really is useful information in terms of the time frame, and it can lead to a proper diagnosis at the end of the day. Level 2 diagnostics includes basic laboratory analysis. So typically, level 2 diagnostics will include histopathology, bacterial culture, parasitology, and maybe some microscopic analysis. For a very long time, histopathology has formed the backbone or the basis for aquatic animal diagnostics. These analyses help to confirm that you have an infectious cause that is causing your problems, either the clinical signs or mortalities on your farm, and it provides strong evidence for diagnosis of the disease. In many cases, some of these tests may also not be specific enough, but you at least know you have a bacterial or a parasite or a viral infectious agent dealing with this. The third level of diagnostics is highly specialized molecular techniques. These techniques include polymerase chain reaction, PCR, and quantitative polymerase chain reaction or real-time PCR, isothermal amplification in terms of lamps, and there are many others that are typically used for diagnostics. These tests generally tend to be highly specific. So in other words, they're used for confirming the specific identity of the pathogen in your sample. They're also good for detecting subclinical infections. In the previous discussion about the purposes for surveillance, we spoke about certifying disease-free and looking at apparently healthy animals. So these are animals that do not have a clinical sign. They appear normal, but they could still be carriers of the pathogen. And PCR sensitivity is really good for detecting these pathogens, or at least pathogen DNA in the sample in subclinical animals. And consequently, this level of diagnosis is very important for supporting international disease reporting or supporting your declaration of your disease status when you're wanting to trade animals from one country to another. To try and bring home some of these concepts, because I accept that they can be slightly abstract without an example. So I'm going to give you run through a quick case study of how a surveillance program was developed in South Africa following the outbreak of a disease which happened in about 2010 and pretty much resolved in about 2015. We still have the odd outbreak, but subsequent to 2015, we haven't had large-scale outbreaks and mass mortalities of animals. And this disease is infection with Haliotissida noduliformans, or ATM, which stands for abalone tubercle mycosis. And the name stems from the tubercle-like lesions that occur on the foot and the mantle of the animal, as you can see on the images on the left. This disease was first noticed through elevated mortalities. So farmers were doing their observations on the farm, and they noticed elevated mortalities. So that was the first level of surveillance, passive surveillance, reporting to a veterinary authority. Their attending veterinarian came out and saw that there was a high correlation between animals with lesions and mortality, and these clinical signs led to the isolation of the pathogen, as you can see on a culture plate on the bottom. We could visualize it in histopathology sections, so that's level 2 diagnostics, and ultimately we developed a real-time PCR assay to detect this pathogen in abalone tissue samples, and that was published in 2012. What this led to was we had a pathogen. Now we needed to determine the spread. One of the reasons for surveillance was to determine the prevalence and the spread of this disease throughout the industry. So this disease has a really distinct clinical presentation. It produces higher mortalities, and it has these typical nodular lesions on the animals. So we designed a stock inspection protocol where we go to all the farms, look at a representative number of animals from the population to ensure that we have at least some statistical confidence in whether the farms are infected or not. And that was the threshold for observations at that stage. If you see all the dots around the coast of the South African coastline, those are where the abalone farms were during that time, all the grow-out areas. We surveyed all of those farms, and we found the blue dots represent farms where we could not find the pathogen. The red dots, or the yellow dots, sorry, are farms where we found clinical signs of the pathogen or elevated mortality, but we couldn't confirm it through PCR. And the red dots are all the sites that we confirmed as positive through PCR. This led to further investigations, and as I mentioned yesterday during the discussion, the disease was really highly correlated to a certain production system or infrastructure, and when the farmers did that or changed that, the disease effectively went away. Subsequent information to that, research has been ongoing. We've discovered, and not only us, but researchers in other countries, they've discovered that this pathogen actually occurs everywhere in the marine environment and is opportunistically pathogenic. So when animals are stressed, if you think about the syndemic pathway again, when any of those other parameters are disturbed, this bug becomes seriously pathogenic and can lead to large-scale mortality. If you look at that top image on the left-hand side, those are just some of the mortalities that were recovered during a stock inspection process from a farm. This process, however, has further utility. We realized that the criteria used for this study is not only indicative of abalone tubercle mycosis. Mortalities or deaths in the population are a clinical sign of many infectious agents. We've modified the stock inspection process. We still go out and look for mortalities, and we look for animals with lesions, but we now also look for some of the clinical signs of other diseases. For example, Brett mentioned yesterday, that the abalone adhere to their substrate quite firmly. So they are quite sticky. If your animals are not adhering, that is showing weakness, and it's a clinical sign that we look for. Abalone, typically, if you put them on their shell, they have a reflex that they'll turn back onto their feet. If that reflex is inactive, then it's a sign of weakness, and that's a clinical sign that we measure. And there are others. So we've now expanded the utility of these farm-based inspections with broader parameters to serve a broader purpose and to increase the utility of this exercise in terms of disease surveillance. The stock inspections are conducted at least once a year on each farm by our department, the Department of Forestry, Fisheries and the Environment, or DFFE, as it stands on the slide. But each farm's private veterinarian also does at least 2 inspections in a year. And I say at least because there are epidemiological information that informs the frequency of surveillance, which I'll get into now. Once again, I think we're out of internet. Merci, Maria. All right, so just to recap, we expanded the purpose of this surveillance program or surveillance activity to survey for more than just abalone tubercle mycosis. As I said, we've got field diagnostic methods that are supported by clinical or laboratory methods. We've expanded from just looking for lesions in that top one to look at other clinical signs like a reduced foot, as you can see in the abalone on the bottom left. And if you really look carefully, it's got a distended proboscis or mouth, which are both clinical signs of 2 different diseases listed by the World Organization for Animal Health. Where possible, we still try and culture the pathogen, or sample for histopathology, and ultimately we take samples for PCR as well. Right. Here we go. So when we're on the farm and we do stock inspections, the first thing we do is we quantify the mortalities in the baskets. So we don't go and look at all the baskets. The farms are just way too big and there's way too many. So we look at a representative sample that gives us 95% statistical confidence that we will find mortalities in a basket in more than 2% if it occurs at more than 2% of the basket. So if there are signs of dead animals in more than 2% of the baskets on the farm, we have enough the sample size is sufficient to give us enough statistical confidence that we'll find it. We classify our mortality data into 2 categories. The one is freshly dead, and the other one is total mortality. So the freshly dead mortality, why we separate that out, is because abalone that die at most will last 3 days in the system, where you can still find an abalone with enough tissue that is still usable for diagnostic testing. So for level 2 or 3 diagnostics. They just decay, they break down too quickly in the environment. They get eaten by other abalone, and there are other organisms in the water that just cause decay. So this actually gives us a unique snapshot into acute mortality. So if you have a high number of fresh mortalities in the sample, then you have fairly good confidence that something is going on. On the other side, we look at total mortality. So any indication of mortality in that basket, either a shell that's left behind or a fresh mortality, will all be added together to determine the total mortality figure. When we look at these data that we've collected now over more than 10 years of stock inspection data, we can see that on the left-hand side, our fresh mortality data is less than 10% of the baskets on all the farms that we've surveyed over 10 years. Less than 10% of the baskets will have fresh mortalities. So this gives us an observation threshold. Anything exceeding that should trigger an alert. The total mortality is slightly different, and this depends largely on how often the basket is handled. Baskets are handled for when they size grading the animals or when they're cleaning the animals. And the frequency at which that occurs will provide a little bit of variation in the total mortality data, or the data for when we still find shells in the baskets. However, having said that, if we look at over the 7 years or 10 years of surveillance on 15 abalone farm sites, we'll find that up to 20% of the baskets that we look at will have some indication of mortality. And we regard that as normal mortality level. And anything above that triggers a flag, and some action is needed. So we've got 3 risk categories identified from this data. Anything below 20% prevalence is a low risk or no further follow-up. Above 20% but lower than 40% we'll regard as a moderate risk. It requires some follow-up action. And then anything above 40% will be regarded as a high risk. So if we view this graphically, we can see that on the green block on the bottom left, you've got your fresh mortalities on the y-axis, and we've represented the total mortality on the x-axis. You'll see that anything up to 20% on the x-axis and anything up to 10% on the y-axis is in that first light green block, and those are regarded as low risk or no. Then we've got everything from 20% live or fresh mortalities. Live mortalities, what am I thinking? 20% fresh mortalities on the y-axis, and up to 40% total mortality represents a moderate risk. And everything above that, outside of those 2 group blocks, is a high mortality or a high risk for mortality. And very often at this stage, we can only assign a risk category. At this stage, we cannot identify what is the ultimate underlying cause for the measurement that we've made. And because of that, it informs the frequency of follow-up surveillance that's required to get to the bottom of the problem. So we developed a decision tree. There we go. We've developed a decision tree for how we deal with these data. So if we have a disease on the farm, we know that ultimately we get the yes on the top, so in that red block, and we implement immediate disease control measures or eradication measures. And generally, depending on the nature of the disease, if it's a WOA listed disease, that farm will be isolated, will stop trading until we can eradicate the disease. If, however, there's no obvious signs of disease, and we've measured that the mortality rate is quite high, it's above your 40% total mortality or above 30% fresh mortality, then we regard that farm as being at a severe risk of either currently expressing a disease or being vulnerable to any pathogen outbreak should that occur. And we're about to lose signal again, I think. It's zooming, yeah, but I still got a slide, so I'll talk to the slide. Oh. 3 slides left. Merci. All right. So if we have a severe rating, if we have such high mortalities that it falls within that severe category, then our action is to reinspect the farm within 3 months. The farmer must take measures to reduce those mortalities, and samples must be submitted to a laboratory for confirmatory testing, or at least to exclude the contribution of high-risk pathogens. If, however, we go onto the farm and we only measure a moderate mortality, or the farm falls into that moderate risk category, then we will only inspect the farm again within 6 months. The risk isn't high enough to warrant immediate action. However, the farmer must still take measures to reduce that mortality rate to what is acceptable. If there's no significant mortality, in other words, the farm falls within that green block on the previous figure, no disease has been detected, there's no significant welfare issues, then we have no further recommendations and we reinspect that farm a year or 12 months after the previous inspection. So the risk-based surveillance is based on the actual risk measured during these events. So in conclusion, there are several barriers that limit the effectiveness of surveillance. And this is a global issue, but it is more prevalent or more prominent in developing countries, and that is limited diagnostic laboratories. For example, the World Organization for Animal Health runs a reference network of reference laboratories, and there are not any reference laboratories for aquatic animal diseases currently on the African continent. Another barrier to the implementation or that limits the effectiveness of surveillance is low awareness of fish diseases. Not many people understand that fish also get sick and they also have pathogens, and these diseases have significant adverse outcomes, which needs to be taken into account. If you don't see the importance of diseases, you're unlikely to report the discovery of sick fish. And that comes down to the next one, which is weak reporting systems. If diseases aren't being reported, if there's no communication regarding what's going on, either between communities, within extension services network, or to the competent authority or veterinary authority, then we have no idea of the diseases that could be threatening our industries. Insufficient funding is a global problem for aquatic animal health services. Veterinarians cost money, but it's always a good idea to, if you have a commercial project up and running and your intention is to increase your aquaculture production, it's always a good idea to have a attending veterinarian who is proficient in aquatic animal health and aquatic animal disease diagnostics associated with your farm. Overcoming these challenges requires investment and capacity building. It requires awareness. It requires extension services. and ultimately diagnostic infrastructure. Every country doesn't need to have a reference laboratory, but it would be good if the African continent or North Africa versus Southern Africa at least have some kind of dedicated diagnostic infrastructure for aquatic animal diseases. So in conclusion, effective aquaculture disease management depends on knowing the pathogens present in the farming systems. This is augmented through farmer observations. You have extension networks. Laboratory diagnostics can add to this. You have national surveillance programs, as the abalone one I've just described. And together these components create a multi-layered early warning system that protects aquaculture production, farmer livelihoods, and international trade. In aquaculture, keeping pathogens under control means keeping them under observation. Thank you for your attention, and my special thank you to your patience for the technical issues, and an even more special thanks to the interpreters who have kindly interpreted my English into French so that you can comprehend what I was telling you. Thank you.
Merci beaucoup, Kevin, pour cette présentation. Non? Oui, allez. Ouais. Comment?
On prend quelques questions. Après, on vous
Alors, donc, on va prendre quelques questions, peut-être 3 questions maximum. Et après cela, on passera à la répartition des groupes pour le travail avant de prendre la pause déjeuner. Oui, est-ce qu'il y a quelqu'un qui veut intervenir?
Je vais commencer par poser une question à Kevin, parce qu'en fait, il a parlé de surveillance ou de détection passive. Et la manière dont il a expliqué, est-ce que cette détection passive-là implique une sensibilisation des aquaculteurs? Est-ce qu'il y a un peu des formations qui sont faites pour les apprendre, les méthodes de détection, n'est-ce pas, de ces maladies-là, ou bien, voilà, c'est leur expérience, ou bien sur les connaissances personnelles qu'ils font ces déclarations-là, quoi. Donc, parce que s'il y a vraiment un accompagnement allant dans ce sens, est-ce qu'on peut parler, n'est-ce pas, de détection passive dans ce cas-là? Parce qu'il y a des efforts qui sont faits pour les former, pour les sensibiliser. Et est-ce que ça c'est fait ou c'est pas fait pour ce type, n'est-ce pas, de détection-là?
Can I continue? So that is a good question, and it has a multi-layered answer. Training and creation of awareness is important at all levels. The training that is provided at an international level is essentially based at improving reporting of, for example, the World Organization for Animal Health listed diseases. The World Organization has a network of focal points in each member country, and at least once every 2 to 3 years, they will present a focal point training workshop in the country for the detection of, well, for implementing the standards and that includes detection of diseases. In addition to that, they've developed, as part of the aquatic animal health strategy, they've developed a very nice online application, and I can circulate the link to that, that helps for the physical detection of all of the listed diseases. So that is available as an extension application. On a similar note, particularly if they are, if it's an internationally relevant disease, or if it is a disease that creates a lot of issues, the Food and Agricultural Organization of the United Nations will also do training. Can be done at the request of a country under a technical cooperation process, and they will then do the implementation. I was personally involved in that type of training way back in 2008, when epizootic ulcerative syndrome broke out in the Zambezi system and subsequently moved south into South Africa. They had really in-depth workshops on what are the clinical signs, reporting lines, who to report to, and so on, and even developed a once-off surveillance program to determine the spread of this disease over a short period of time. I know, as in addition to all of this, the African Union, AU-IBAR, they also have just launched a new aquatic animal health strategy, and possibly through that avenue, there could be training as well. At the farmer level, that comes down to the veterinary authority, and mostly led through either universities or government extension services, where the importance of diseases is training should be provided to create the awareness of the importance of diseases and how to recognize them. Not every farmer has to be a veterinarian. Not every farmer can perform diagnostics at level 3 as I presented. But it's really important that every farmer understands what signs are typical of a healthy fish and how to recognize sick fish. And that's the first step. And the second step is, who do they report it to? What are the support services in the country for that to occur? And that's why I said in my last slide, one of the major constraints to aquatic animal biosecurity is awareness and capacity building. So training events like this hopefully have helped to create some awareness, but definitely as an industry grows, generally this kind of interest in this kind of subject matter will grow with it.
Merci beaucoup. Est-ce qu'on a un autre intervenant? Non? Oui, Papa Amadou.
Alors, bonsoir et merci encore une fois, Kevin, pour la qualité de la présentation. Alors, ma question en est que, par exemple, il a un peu parlé de cette gestion collégiale-là. Par exemple, les réseaux des exploitants, comment ils parviennent en tout cas à faire ces gestions-là qu'il faut. Bon, ma question à savoir, par exemple, si on essayait de répliquer ce modèle-là au niveau local, sur quel levier basé? Est-ce que c'est l'État qui essaie en tout cas de renforcer cette entente autour de ces exploitations, ou bien ce sont les exploitants eux-mêmes qui se regroupent en, comment ça s'appelle, en coopérative pour prendre, voilà, pour faire la gestion collégiale? Merci beaucoup.
Thank you. I think that is a question that's relevant to many countries. In many countries, the creation of awareness is not only at the farmer level, but the policymakers and politicians and senior officials need to be educated about the importance of aquatic animal health as well. And in many countries, that doesn't take place. So cooperative surveillance and cooperative animal health management is probably in small industries best managed between farmers. You know how important your fish and your fish health is to you. Your neighbor will have the same understanding, and sharing amongst each other information and capacity building and awareness about potential disease risks and threats, I think should first happen. It should first you get buy-in from the farmers. It shouldn't be something that is forced on you from a government official, because at the end of the day, it's the farmer's livelihood and those people working on the farm that depend on the farm's success. So to me, it's a bottom-up process. You should have coordination between farmers, and that should feed up and hopefully eventually to political level.
Madame Sall.
Bonjour. Merci beaucoup, Monsieur Kevin, pour la brillante présentation. En fait, je vais un peu rebondir sur ce que Amadou a dit. Juste pour dire qu'actuellement, le Sénégal, il y a déjà un réseau qui constitue les producteurs. Donc, ils se connaissent pour la plupart. Parfois même, je me dis que si un problème se déclarait, je pense que la quasi-totalité des fermes seraient touchées parce qu'ils ont une étroite relation entre eux, soit des échanges d'alevins ou de géniteurs entre eux. Même si deux fermes ne se connaissent pas, il y a une troisième ferme entre eux qui se connaissent et qui ont des relations entre eux. Maintenant, le souci, c'est que, comme vous l'avez dit, il n'y a pas de réseau de laboratoire. Donc, les problèmes ne sont pas très connus dans un niveau très élevé. Par exemple, les maladies, parfois on en remarque, mais des diagnostics très poussés ne sont pas du tout. Et également, parfois, on remarque que certaines exploitations rencontrent des problèmes, mais ne veulent pas le dire. Peut-être parce que ils ont peur ou bien ne veulent pas se faire voir comme une ferme qui a des problèmes. Et pourtant, c'est des exploitations pour la plupart qui sont dirigées par des gens qui ont des niveaux d'études élevés, même en pisciculture, qui ont fait des formations en pisciculture. Maintenant, je me demande, vu que si à chaque fois ils font ça, ne déclarent pas leurs problèmes, essayent de le résoudre en interne, et pourtant échangent des matériaux ou bien des individus avec les autres fermes, qu'est-ce qu'on peut faire réellement pour remédier à cela? Parce qu'à un moment donné, ça risque d'exploser et d'être un problème très, très grand. Voilà, merci.
Thank you for that question. I think the question to me is a compliment because it suggests to me that you've understood a lot of what we spoke about yesterday. So thank you for that. I think your question is a difficult one to answer. It is true in all spheres that farmers, particularly if they have a financial incentive to sell their stock, might be reluctant to report disease outcomes because they want to sell animals. They don't want to be stopped from selling animals. I think the awareness needs to go beyond that. It's not just about the impact that you have on yourself, but if I buy sick fish from somebody today and it causes me great financial loss, I'm not likely to buy from that same person tomorrow. So it's really important to understand, particularly in institutions where there's a lot of change and exchange of livestock between farmers, there really needs to be a system of integrity and trust, because your reputation can create far more damage down the line than protecting a certain cohort or batch in the short term. I know it's a difficult thing and it's something that you can't necessarily control, which is why it is also listed as one of the weaknesses for passive surveillance kind of reporting. But yeah, excuse me, even at the international level, the standard of operation is one of trust and integrity. So you would be unlikely to buy fish from a farm or a producer or a hatchery that has a lower health status than what you have on your farm. So, and that's the reason why farm level coordination is important. You may be implementing biosecurity measures on your farm to increase the quality of your fish. But if you're still buying fish from somebody who doesn't have those same values, then your work and your efforts are in vain. So building that trust, creating that common awareness and integrity amongst an industry, I think is fundamental to the success of surveillance programs.
Merci beaucoup, Kevin, pour ces éclaircissements. Donc, on prend dernier intervenant, s'il y en a. OK. Jab Bye. Madame Nyinga.
Bonjour. Merci, Monsieur Kevin, pour la belle présentation. Bon, moi, je vais partager mon expérience en ferme et lui poser une question par rapport à ça. Parce que dans notre ferme, on avait des tilapias, des super mâles qui nous étaient venus de la Suède, qu'on avait élevés depuis toute petite et qu'on voulait avoir une population, une descendance toute mâle en tilapia. Alors, lorsque les poissons ont atteint une poids de 300 à 400 grammes, il nous est arrivé que dans la ferme, on assistait tous les jours à des mortalités et à des changements de comportement vis-à-vis des poissons. Mais les mortalités n'attaquaient que les mâles, que les super mâles. Ce qui nous a un peu en tout cas, c'était notre problème et on a tout fait. En tout cas, moi, j'ai beaucoup creusé. J'ai partagé avec des personnes ressources. J'ai creusé sur l'Internet. J'ai des images avec moi. Le problème en est que la maladie n'attaquait que les mâles, les super mâles. Et quand on faisait les déplacements pour nettoyer les bassins, la maladie les suivait dans les autres bassins. Et les femelles restaient toujours intactes. Les mâles avaient les yeux gonflés, le ventre gonflé, les écailles, ils se désécaillaient carrément. Et il y avait aussi des oui, j'ai les images avec moi. Et ma question, c'était, par rapport à votre expérience, qu'est-ce que vous auriez me conseillé sur ça? Une personne ressource m'avait conseillé de faire la solution saline et de le tremper pendant 30 minutes. Et lorsqu'on faisait ça, soit on guérissait le poisson et la maladie attaquait les autres, soit on ne guérissait pas. On ne sait pas et le problème ne s'est toujours pas résolu jusqu'à présent. Merci.
Thank you very much. That is, once again, unfortunately, one of the Okay, so this fish that I'm looking at on this video clip has a distended belly, severely. Its scales are raised. It resembles a pine cone, a pineapple, and the eyes are bulging. So those are clinical signs, typical clinical signs. That bulge in the stomach is caused by fluid retention. And so there's a lot of liquid in that abdomen. And typically, these kinds of infections are caused, or these kinds of clinical signs rather, are caused through some disruption of kidney function. Most often, it's bacterial and can cause a condition called dropsy. But there are other causes too. So I think the suggestion for salt was probably a good first aid remedy, but what you really require is a diagnostic that can identify whether it is in fact bacterial, and a veterinarian who can prescribe the appropriate medication. Indiscriminate use of antibiotics or prophylactic use of antibiotics leads to other downstream complications. So it's really important that the diagnosis is sound. The advice I would give to you is through observations, you should isolate infected fish. The best way probably to do that is to move the uninfected fish into a clean system so that you can disinfect and treat the current fish. Generally, salt is a good, in freshwater fish, salt is a good remedy. Tilapia generally have a high salt tolerance, so you could probably even extend the duration of the treatments a little bit. But the microorganisms and parasites typically associated with the external surfaces of the organism will be highly susceptible to changes in salinity, and so it will provide temporary relief. I think the importance here is also just disinfecting the system, because the source of infection is somewhere in the system. So unfortunately, this is obviously something that's already happened, but observation, as I've just highlighted in my presentation, and early action. When you see one or two fish with these symptoms, you remove them from the system and take other remedial action to improve the situation. Otherwise, if left untreated, this will spread to all the fish in the tank.
Okay. Oui. Vas-y.
Je vais compléter un peu la réponse de Kevin. Et je vous pose une question. Vous donnez de l'aliment composé, j'imagine, aux poissons. Vous donnez de l'aliment aux poissons. Et comment vous stockez cet aliment-là? C'est à quelle température? C'est à température ambiante? C'est au frigo? Pardon?
L'aliment était conservé dans un magasin fermé.
Un magasin?
Oui.
Je vous dis ça parce que, en fait, nous avons eu une situation similaire avec un poisson chat qu'on cultivait en Amazonie péruvienne. Donc, c'est chaud, c'est tropical. Et on avait ces soucis-là, des gonflements du ventre. On ouvrait le ventre et sortait plein de liquide, comme disait Kevin. Et on s'est rendu compte, après avoir fait tout ce que vous avez fait, donc nettoyer les systèmes, séparer les malades de ceux qui n'étaient pas malades, et ça continuait. Et on s'est rendu compte qu'en fait, l'aliment venait du magasin contaminé. Donc, je vous suggère de voir s'il y a peut-être une contamination dans l'aliment qui fait que vous avez toujours ce problème de contamination. Je ne sais pas ce que c'est dans votre cas. Kevin suggère bactérienne, c'est possible. Ça peut être aussi de champignons. Je ne sais pas si ce sont le même, par contre, le même.
D'accord.
Et
Pour ça, je vous rassure, l'aliment n'était pas contaminé parce qu'on contrôle souvent le magasin et c'est nettoyé de temps en temps. Et même les bassins sont contrôlés. Je prenais les paramètres physico-chimiques tous les jours, le matin et le soir. Le pH, la salinité, la conductibilité, l'oxygène dissous, la température et les nitrites, ammoniac et nitrate, tout ça, on le prenait chaque jour, tous les matins et le soir. Et les bassins étaient renouvelés tous les matins. Pas complètement, mais juste les 15% étaient vidés et encore, on mettait de l'eau encore dans les bassins. Donc, et ce qui me pose problème, c'est que la maladie n'attaquait que les mâles. On n'avait pas de mortalité sur les femelles. Ça n'attaquait que les mâles. Et le problème continue à persister. On n'a toujours pas de solution. Oui, les femelles et les mâles viennent de la Suède, oui. Et c'était des super mâles aussi.
C'était
Des super mâles, oui.
Bon, il n'y a pas une résistance, différence de résistance parce qu'ils viennent de la même zone. Ça, c'est un peu inquiétant. Est-ce que vous les avez mélangés dans le même bac avec les souches?
Oui, dans le même bassin.
Et la souche locale n'est pas affectée?
Non, non. Non, non. Parce qu'on n'a pas ce problème avec les gris, les tilapia gris. Il y avait aussi des super mâles gris venus aussi de la Suède. Ils sont venus en même temps, les rouges et les gris. Mais le problème, c'était avec les rouges et tout le temps, c'était avec les super mâles.
Non, mais si c'est spécifique aux femelles, bon, pas aux mâles, parce qu'en fait, voilà, peut-être ils sont du même karyotype, mais les autres sont femelles. C'est ça, n'est-ce pas? Maintenant, sur le plan éthique, je ne vois pas, voilà, comment ça peut s'expliquer, quoi, franchement. Il n'y a pas de réponse immédiate.
Alors, je pense que pour ce cas, il faudra peut-être adopter les conseils de Kevin, aller voir un spécialiste, prendre des échantillons et faire ses analyses pour voir est-ce que réellement c'est dû à une bactérie ou bien d'autres voilà, micro-organismes qui peuvent être nuisibles. Voilà. Voilà. Alors sur ce, alors, avant qu'on prenne la pause, je vais passer la main à Maria pour qu'elle puisse faire la répartition des groupes et revenir sur le travail qui les attend après la pause. Voilà.
En fait, j'avais plus ou moins dit ça avant, mais c'est vrai que ah, d'accord, ok. Donc, cet après-midi, après ma présentation, qui sera la dernière du cours de formation, on va vous grouper en 4 groupes de 10, 11 personnes, en fonction de combien vous êtes. Ma mère m'a conseillé que vous soyez ceux qui se groupent en fonction de votre affinité. Donc voilà, je vous demande, s'il vous plaît, de vous grouper en 4 groupes de 10, 11 personnes. Et comme ça, au retour, on fera, vous avez eu tous ces petites feuilles avec l'activité qu'on va faire après. Donc, comme je disais avant, vous allez essayer de mettre en pratique tout ce tout ce que vous auriez entendu parler pendant ces 2 jours-là. Et puis aussi utiliser votre sens commun, votre intuition. Et comme ça, vous allez à la fin, créer un petit dessin des fermes aquacoles avec les espèces que vous auriez choisies et vous aurez pris en compte tous les aspects qu'on avait mis dans cette feuille-là, au niveau des espèces, en matière de biosécurité, des nutritions, de transfert des nutriments à travers les systèmes, etc. Chacun des groupes peut choisir une personne qui va être la personne qui va exposer ces systèmes-là. On va placer une grande feuille au mur et on aura ces 4 systèmes d'élevage et on va en discuter un après l'autre et comme ça on apprend un tout petit peu. On vous met dans l'esprit plus pratique. D'accord?
OK. Donc, c'est bon, on va prendre la pause. Donc, on reprend à 14h30, hein? 14h30. Je pense que l'idéal, Maria, je pense que l'idéal, c'est de répartir d'abord les groupes avant la pause. Comme ça, au retour, ouais, voilà, après. OK, d'accord, d'accord. Donc, déjà présent, vous pouvez commencer à choisir, voilà, là où vous voulez être, quoi. OK. ba sànqay doog a remarqué que ne témoin bi muy fi témoin bi remarqué mu ko donc d'habitude man yi may gis mooy c'est beaucoup plus éclairé quoi fii nii quoi mais témoin bi léegi laa koy doog a seetlu man fii laa doon xool ba xam nga tàkk na tàkk yéen bëgg a taal quoi ah man de bi ma toog nii ni ma fi jógee ndax dafa wax rek nee na da ngeen doon def ko man ko tamit demul bi laa dee dañu ko doon moo jaaxal yow ana ko doon leen bi Sama pengen ambil itu.